DOE CSGF Fellow Kayla McHugh in the field of computational and systems biology at the Massachusetts Institute of Technology.
Hi everyone. So I wanted to take this opportunity today to talk about my work modeling pre-mRNA splicing with the stochastic grammar framework. But before I get into the details of that, I wanted to start with just a little bit of background. In particular, I wanted to start by telling you about the central dogma of molecular biology, which states that the biological information for a cell is stored in the DNA, which is transcribed into RNA, and then finally translated into protein. While, as with most things in biology, the truth is a lot more complicated than that, this is an important information pathway in the cell. However, this simple description somewhat elides the role that RNA, in particular messenger RNA or mRNA, plays as an important regulatory step. I've illustrated here a slightly more detailed version to show how DNA is directly transcribed into pre-mRNA, which then undergoes several processing steps before being translated into protein. The particular processing steps that I've highlighted here are polyadenylation, 5' end capping, and pre-mRNA splicing. And as you may have guessed from the title of this talk, the particular step that I'd like to describe further is splicing. So eukaryotic genes have this interesting feature where the sequence that encodes a particular protein is not always stored in one contiguous chunk in the DNA. Instead, they are interrupted by interstitial sequences known as introns. These intronic sequences are transcribed into the pre-mRNA and then removed or spliced out. The remaining sequences are known as exons, and they make up the mature mRNA that will eventually be translated into protein. The boundaries between introns and exons are known as splice sites. In particular, the boundary with the exon on the left and the intron on the right in this schematic is called a 5' splice site, and the boundary with the exon on the right and the intron on the left is called a 3' splice site. The molecular machinery that facilitates the splicing reaction is called the spliceosome, and it assembles stepwise on the pre-mRNA transcript, recognizing motifs that mark the splice sites. Once the splice sites are recognized, the spliceosome undergoes several compositional and conformational changes before eventually bringing together the ends of the exons to be joined. An interesting quirk of the spliceosome in many higher eukaryotes, such as humans, is that the initial mode of recognition is thought to be predominantly cross-exon rather than cross-intron. There is then a rearrangement step for the spliceosome in order for it to form across the intron and remove it. A common theory as to why this occurs has to do with the relative lengths of introns and exons. In humans, for instance, most exons are between 50 and 250 bases, whereas introns can be tens of thousands of bases in length. From this, we have this conception that exons represent the key structural element in human pre-mRNA splicing. The components of the spliceosome are not the only factors that are important in determining the final splicing pattern, however. There are also what are known as splicing regulatory elements, or SREs, which can either enhance or suppress splicing in the local sequence by encouraging or discouraging the recruitment of spliceosomal components. SREs play an important role in distinguishing true splice sites from the many pseudo-splice sites that exist throughout the human genome. While SREs are known to be vital to the splicing process, they are not as well understood as the motifs that mark the 5' and 3' splice sites, and improving our understanding of them could also improve our ability to understand and treat splicing-related genetic disorders. Additionally, different transcripts of the same sequence can have different splicing outcomes, which is known as alternative splicing. One of the most common and well-studied types of alternative splicing is exon skipping, where an exon is either included or excluded from the final transcript wholesale. In this case, we can define a % spliced in, or psi value for this exon, which represents the fraction of transcripts in which the exon is included. A related concept is that of exonization, where an intronic sequence has some potential to be used as an exon, but never is. This sequence can then be exonized by mutations as part of the process of evolution, or by outside perturbations, such as the introduction of antisense oligos or small molecules, or by changes in trans factor concentrations. Evolutionarily speaking, the majority of these exonization events result in frame shifts and premature stop codons, disrupting the function of the protein or triggering the nonsense-mediated decay pathway. In all likelihood, this would also be true of exonization via the interventions I just mentioned. However, this could be a desired and beneficial outcome in the case where one is interested in downregulating or silencing the expression of a problematic gene. Now, the last concept I want to touch on in this sort of background portion of this talk is stochastic context-free grammars, or SCFGs. And let me tell you, being a CSGF fellow studying SCFGs has been a bit of a tongue twister. But in any case, what an SCFG is at its core is a probabilistic string generation process in much the same way that a hidden Markov model is. Indeed, SCFGs are actually a superset of HMMs, which allows for a loose analogy to be drawn between the component parts of the two. The SCFG's terminals are analogous to the HMM's emissions, the non-terminals to the hidden states, the rules to the hidden structure, and the probability distributions over those rules to the emission and transition probabilities. Now, before I talk about the particular grammar that I've developed, I want to first briefly discuss how it fits into the overall structure of my model. A general overview of how I conceive of it is shown in this flowchart here, where I start with a set of annotated RNA sequences and then independently extract information about the splice sites and splicing regulatory elements. For the splice sites, I use the splice site maxent scan method, which is commonly used in the field, and for the splicing regulatory elements, I use the ratio of their frequencies in internal exons and introns, which has been used in the literature. Then, once I have that information, I combine it in order to create a preprocessed sequence that I can use to determine parameters for my SCFG based on the splicing pattern annotations for the sequences. Having learned the parameters on a training set, I can then run a test set of sequences through the same preprocessing procedure and feed the resulting preprocessed sequence through the algorithms for my trained grammar. My particular grammar is shown here, and to illustrate it, I have cribbed liberally from the tradition of hidden Markov models because I think it does a good job of representing the underlying structure and behavior of the grammar. One difference I want to highlight is the presence of these curly braces floating above and below some of the non-terminals. These are there to indicate the non-geometric length distributions that using an SCFG allows me to incorporate. The learned versions of these distributions are shown in these plots to the right. To further illustrate the behavior of this grammar, I want to briefly go through it in a generative sense. Starting with the single non-terminal, this is the traditional way to start the generation process, we can look at our S non-terminal in our diagram here and see that there are two different options. We could take this arrow on the right with probability PME, or with probability P1E, we can take this arrow on the left to this ES state. So here, we are in an exonic state with the subscript S indicating that we are in a single exon gene, that is, a gene that has no introns. Looking above this state, we sample from the associated length distribution to get a value D for the length of the exon. Then we emit D exonic bases to complete the generation of this exon. Following that, we take the only outward arrow with the probability of 1 to this epsilon state, which represents the end of our generation process and is essentially a way to get around some of the technicalities of what makes a grammar context-free. Once we've reached this state, we're done. But if we were to rewind back to the beginning, we can see what happens if we instead take this arrow to the right with probability PME. In this case, we end up in the state EF, another exonic state, but this subscript indicates that it is the first exon in a multi-exon gene. As with the single exon case, we sample from its length distribution to get a value D, and then emit D exonic bases to form the first exon. Following that, we take the only outward arrow to this 5' splice site state, which has a single emission before continuing on to the intronic state. We sample from the intronic length distribution and then emit however many intronic bases the sampled value dictates. Again, we only have a single outward option, which takes us to the 3' splice site state. This state also only has a single emission, and then we are faced with another choice. We could take this lower arrow with probability PEO, or with probability PEE, we can take this upper arrow to end up in this EM state. This is another exonic state, the subscript M here indicating that it is a middle or internal exon. As before, we sample from its length distribution and then emit however many exonic bases. Following that, we're back at the 5' splice site state with its single emission, and at this point, the behavior is the same as before. We generate an intron, and then a 3' splice site, and then we are back to being faced with a choice. We can loop through this little cycle as many times as we want, each time with probability PEE, generating a new internal exon and intron each time. But eventually, we will end up in this 3' splice site state, and with probability PEO, we will take this other arrow to end up in the EL, or last exon state. We sample from its length distribution, emit however many exonic bases, and then end up back in our end of generation state. Now, similarly to how this representation is based in the HMM tradition, and I was able to draw the earlier analogy between components of HMMs and SCFGs, I can also define analogous algorithms to the canonical algorithms of an HMM for my SCFG. For SCFGs in general, one would typically consider the CYK inside and outside algorithms whose computational complexities are cubic in order in terms of the length of the sequences, but because of the way my grammar is structured specifically, I can define more directly analogous algorithms that are only quadratic in order, in essence, defining Viterbi forward and backward variables. With the Viterbi variable in particular, I can find the most likely splicing patterns for the transcripts in my test set and see how well they do at capturing the exons. To start out with, I considered a subset of my overall model, excluding the splicing regulatory elements and using only the splice site and length distribution information. With this, I ended up with a sensitivity of 23% and a precision of 58%, reflecting the fact that my model does a better job of selecting true exons than it does of selecting enough exons. Now, these numbers are obviously not ideal, but they are on par with, if not slightly better than, other existing models that use only splice site and length information, so it's about the level that I expected. For another test, I included all hexamers, so all possible sequences of length 6, as splicing regulatory elements. And with this, I see a slight increase in my precision and a more substantial increase in my sensitivity. But while this increase is non-trivial, it's still not to the level I'd like it to be. And one potential reason for that is the way that I'm including splicing regulatory elements. As I discussed before, I'm using a frequency ratio of frequency in internal exons and frequency in introns, which a paper that I was partially inspired by calls a reasonable default, but says that it may not be optimal. And that has to do with the fact that these scores are based on the way that SREs are expected to be distributed in mRNA sequences, rather than being learned from information about their behavior as SREs specifically. But with my framework, this is something I could potentially accomplish. So to that end, one of the things that I've been working on recently is developing and implementing a method by which to learn new SRE parameters and thereby potentially learn something new about their biological function. In terms of parameter learning for SCFGs, the sort of canonical example is maximum likelihood estimation through expectation maximization, so akin to Baum-Welch. However, this is generally used in the case of unlabeled sequence data, which is not the situation that I have. Instead, I was inspired by one of the concepts that I discussed earlier of percent spliced in. For a particular exon, we can determine its psi value, that is, how frequently it's included in transcripts. And then we can define this function phi as a probabilistic description of the psi value for internal exons. This probabilistic expression can be written in terms of the forward and backward variables that I showed before, along with a few other parameters of the model. Now with this expression, I've developed a gradient descent approach to learn parameters for the SCFG SREs specifically by minimizing the cross entropy between phi and psi that I'm currently working on the implementation of. At the same time, I'm also exploring another application of this model and of this phi expression in particular. As I can determine phi for any subsequence, not just known internal exons, it provides me a way to investigate potential exonization events. I've included here a plot for a particular gene in order to illustrate this further. In the bottom row here, you can see the annotated experimentally observed splicing pattern for the gene, a first exon, 2 internal exons, and then a last exon. In the next row up, I have the Viterbi most likely splicing pattern, which in this case matches the annotated splicing pattern exactly. And then above that, I have all the regions in the transcript with a phi value of greater than 0.05, with their height determined by their probability. As you can see, the 2 annotated internal exons have the largest phi values, and the first internal exon in particular has the highest phi. Alongside that, there are several regions of lower phi that are identified, mostly within the last intron, some of which are overlapping. The one that I've boxed here in particular has the largest phi value of all the non-annotated exons, and so might represent the strongest target for an exonization event. One of the potential outcomes of this analysis, if done on medically relevant genes, is the identification of exonization events that represent new therapeutic targets, which is one of the things that I'm excited about going forward. And with that, I would like to take just a quick second to extend a thank you to the members of my lab, the Computational and Systems Biology program at MIT, and the DOE CSGF for all their support, and also to you for taking the time to listen to this talk. Thanks!